So in regards to differentiating between host range and viral specificity, again, host range deals with what specific host a virus can infect. And then assuming that the virus is in fact in the right host, viral specificity refers to specifically what types of cells the virus is going to be able to infect or bind to. So with viruses, they need to make it to the right host and they need to make it to the right cell. If they're able to do so, then their spikes will match the receptors on the host cell membrane, and that's going to facilitate the virus being able to penetrate the host cell. So assuming the spikes do in fact match the receptors, the next step is penetration. Penetration is when the virus is going to enter the host cell. This can happen through two different mechanisms depending on the type of virus, one being endocytosis and the other being membrane fusion. All viruses have the ability to enter through endocytosis, which is what we're seeing here in A. So for that to happen, essentially a virus would attach its spikes to the receptors on the host cell membrane. The host cell, which for example would be the human cell, would then think that it had just bound to some type of food or nutrient, so it would engage endocytosis, which is basically cellular eating. What's going to happen is because the host cell thinks it has just bound to a nutrient, it's going to extend its cell membrane and engulf the entire virus, much like it would food. Now, at this point, the entire virus is inside the host cell, so the capsid, potentially even the envelope, if it was an envelope virus, and the nucleic acid core with the genetic information, the entire virus at this point is inside the host cell. Another mechanism of penetration is membrane fusion. Now, membrane fusion can only be used by envelope viruses, which will make sense here in a second. So for membrane fusion, what's going to happen is the envelope virus is going to have already bound to the surface of the host cell, so the spikes are going to be attached to the receptors. And then what's going to happen is the envelope of the virus is actually going to fuse with the cell membrane of the host cell, and the envelope is actually going to stay out in the cell membrane of the host cell, and the only part of the virus that's going to enter is the capsid and the genetic material. But again, that envelope is actually going to stay out here in the cell membrane, and that's why only envelope viruses can do membrane fusion, because only envelope viruses have an envelope that's able to fuse with the cell membrane of the host cell and then allow the rest of the virus, which in this case would be the capsid and the nucleic acid core, to enter the host cell. So definitely want to make sure you understand the two different types of penetration, endocytosis being something that all viruses are capable of engaging in, and membrane fusion being something that only envelope viruses are capable of. Regardless of whether a virus penetrates the host cell through endocytosis or membrane fusion, uncoding is always a necessary step with animal viruses. So once a virus enters the host cell, or penetrates the host cell, its outer layers need to be taken off. So if that's a naked virus, it's going to be the capsid. If it's an envelope virus that has entered in through endocytosis, it's going to be the capsid and the envelope that needs to be taken off. So basically what happens during the uncoding stage, to take off these outer layers, the host cell is going to release digestive enzymes that is going to break down the capsid and potentially even the envelope. It's going to break down those outer layers and free the genetic material of the virus. So the DNA or RNA would be freed from the capsid at this point, and then that genetic material is going to head to the nucleus of the host cell and start the process of shutting down and manipulating the host cell. So regardless of how a virus penetrates the cell, it needs to be uncoded. really to take off any of those additional layers, especially the capsid, to free the genetic material. I kind of always think about it as going into someone's home and getting comfortable. You're going to take off your coat. A virus has just entered its kind of new home. It needs to make itself comfortable and go through uncoating. So it's going to, the digestive enzymes are going to take off the protective layers, such as the capsid, to free the virus's genetic material so it can make itself at home, essentially, by taking over the host cell. So at that point, the genetic material of the virus would essentially shut down the host cell and manipulate the host cell into producing more viruses. So we get to the synthesis stage. During the synthesis stage, the host cell is going to synthesize the viral parts. So if it's a DNA virus, the host cell is going to start replicating thousands and thousands and thousands of copies of viral DNA for all the new viruses. It's also going to need to make viral proteins. So the host cell is going to start manufacturing all the viral proteins, for example, the capsomeres that will make the capsid and those spikes that will eventually be used for attachment. But the primary thing that is happening during the synthesis stage is the host cell is now synthesizing the parts of the virus. So it's producing more DNA or RNA, depending on what type of virus we're talking about, and more viral proteins. Step 5 is assembly. So if we make all the parts in the synthesis stage, in the assembly stage, we need to put all the parts together to make mature viruses. And it's going to be a lot of mature viruses. Thousands to potential even 1 million viruses are going to be put together inside of this host cell. So typically what happens is the capsomeres start forming around the genetic material, like the DNA, to form the capsid. And then if the virus is meant to be a naked virus, then the spikes would just attach there to the capsid. But if it were to be an envelope virus, the spikes need to eventually be on the envelope. So the spikes, instead of getting attached to the capsid, would actually get attached out here in the cell membrane. And then eventually, when the virus leaves the host cell, it would pick up that cell membrane with the spikes attached. But the main thing to remember in the assembly stage is this is when we're putting together the parts of the virus to make the mature virons. So again, that's just going to be forming the capsid around the genetic material. And then, again, if it's a naked virus, the spikes would get attached to the capsid. If it's an envelope virus, though, the spikes are temporarily sent out to the host cell membrane where they'll be retrieved in the next step. So the last step of the process is release. So this is when the viruses, the mature viruses that we've just created, are going to leave the host cell. And it's going to be a lot of them. Like I said, for something like the influenza virus, one goes in, but potentially 1 million come out. Something like polio, one goes in, 100,000 go out. So we're talking pretty substantial numbers here. So viral host cell release can happen in one of two ways depending on what type of virus we have. If it's a naked virus, meaning it only has a capsid, does not have an envelope, if it's a naked virus, the host cell is going to get so full with viruses that it just pops like a balloon, and the viruses are released through cell lysis. So just imagine a balloon just expanding and expanding, but those are all the new viruses in there. And then at one point that cell just can't handle anymore, and it bursts through cell lysis. That obviously kills the host cell, but it releases thousands and thousands of viruses into the body that will then use their spikes to go attach to new host cells, and the whole cycle just starts all over again. So that is how naked viruses are released from a host cell. It's different with envelope viruses, though. Envelope viruses cannot leave through cell lysis. If a cell bursts through cell lysis, the cell membrane is destroyed. And with envelope viruses, they need to take a piece of the cell membrane with them as their envelope. So they can't just blow up a cell or the cell membrane goes away and then they have no way to get their envelope. So envelope viruses always release through a process known as budding, which is actually easier to see in this previous image. So once the virus is mature, it actually travels right underneath the inside of the cell membrane, and it's actually going to push on the cell membrane and eventually escape, taking a piece of the cell membrane with it as its envelope. And it's very important that the spikes were already present there from the assembly stage previously, so that when it takes a piece of the cell membrane with it as its envelope, it's going to also get its spikes. If this virus forgets to put its spikes out here in the cell membrane during the assembly stage, and it just leaves and takes the cell membrane with it as its envelope, but there's no spikes present, that's a defective virus. That virus doesn't have any keys, it doesn't have any spikes, so it's not going to be able to attach to a new cell. So really important that in envelope viruses, the spikes are sent out to the cell membrane during the assembly stage. So when these viruses leave through budding and take the cell membrane with them as their envelope, the spikes are going to be present. And we can see this here, what this looks like in real life. So this is a cell actively infected with a virus. All the little dark staining regions throughout are mature viruses. They're all heading to the cell membrane, where they are going to bind to the inside of the cell membrane and push outward so that they can take a piece of the cell membrane with them as their envelope. So here we can see a mature virus that has in fact taken a section of the cell membrane with it as its envelope. So envelope viruses will always be released through budding to take a piece of the cell membrane with them as their envelope. Whereas naked viruses that don't need an envelope, they're just going to leave through cell lysis, so the cell is going to burst. This is a really good video that I do recommend you watch on your own time. It goes through all the stages that we just talked about. This specific video goes through the replication cycle of an envelope virus that's infecting a human cell, but you get to see absorption and uncoding and synthesis, assembly, and then even release through budding. So this is definitely something that kind of just ties everything that we talked about together. So when viruses infect a cell, they obviously damage the cell, and any type of damage that we can see under the microscope is actually going to be called a cytopathic effect. So if we look at a healthy cell and a cell that's infected with a virus under the microscope, they're going to look different. And any differences that we can see under the microscope within the virus-infected cell, those are going to be called cytopathic effects. There's some specific examples of cytopathic effects, not things that you need to memorize. I'm just more concerned that you know what a cytopathic effect is. But sometimes the cell's size or shape may change. So for example, if a cell is infected with 1 million viruses, it often will appear larger. It's expanding to accommodate, all the new viruses. So often the cell's size can change considerably. As the virus infects the host cell, it does create some damage to the internal organelles, like the nucleus, the Golgi apparatus. So the internal organelles may stain differently because of damage that we're seeing. Some viruses, when they infect cells, cause the cells to fuse together and form this mass of infected cells called a syncytia. That's common for the respiratory syncytial virus or the RSV virus. So any type of these changes that cause a cell to appear differently under the microscope when infected with a virus would be known as cytopathic effects.